All right, hopefully that's not, whoa. Hey, it's working, yeah. Maybe I'll move this down a little bit. Does that feel about right? Great, well, it's a pleasure to be here, and I'm excited for a number of potential collaborations with UW and with the eScience Institute in particular, and just kind of a lot of great discussions there. So I'm going to tell you about what we're doing at ISB and explain what I mean by these personal dense dynamic data clouds that I've got here in the title. So I'll just start with disclosures, various companies and things I'm involved in. The main one here being a company called Aerovale, which I was a co-founder of along with Lee Hood and Clayton Lewis. And they helped fund one of the studies that I will talk about today. And we do have a partnership with them, which I'll explain as we go along the way. Okay. So I'll start here. This is a person who is probably familiar to a number of you. This is Larry Smarr. This is a picture of him from the cover of Technology Review, MIT's magazine. And Larry is kind of either the dream or the nightmare of the patient of the future, depending on kind of your point of view and what kind of doctor maybe is thinking about this. But how many of you know who Larry is? Is this, I know, yeah, okay. So Larry is the head of the San Diego Supercomputing Center, and he's also become quite well known as one of the most phenotyped people in the world. So I gave a talk a little while ago at UCSD, and you can go in, and he has a basically big monitor. It's about the size of the side of this room here, you know, and it basically displaying nothing but data about his body. And Larry is Larry's a very open person. If you go to his talks, he'll hand you out a 3D printed version of his diseased colon. So he likes to do this kind of thing. But he had an interesting experience a few years ago, which is he'd been monitoring lots of aspects of his body. And so at one point, he had a number of markers that just jumped to a scale that was radically different than anything he'd ever seen before. And he'd been monitoring himself on a daily to weekly basis for 10 to 15 years. And so when this happened, he talked to some of his biology colleagues. He went into the doctor and he said, I think I'm getting inflammatory bowel disease. And his doctor said, how do you feel? He says, I feel fine. And he says, but look at these data. He says, well, if you feel fine, there's nothing we can do. So he went to one doctor, he got that response, went to another, got kind of the same thing. So he goes home. A few months later, he ends up having an acute episode. He gets taken into the emergency room, and what does he have in this case? IBD. Now, that's an anecdote. Could have gone differently, right? Could have been wrong. But I think that story is evocative of the kinds of signals that may exist in the body that are related to disease before it happens that we don't know about because we don't usually monitor for them. So what we set out to do was to take Larry's experience and try to generalize that to many, many people and try to build a science out of this monitoring for early stage disease. So another thing I wanted to set up before we dive into data is this notion of imprecision medicine. This is a slide taken from Nick Shork from a great paper that he wrote in Nature. And basically, and maybe this slide is familiar to some of you, but shown there in the purple, well, just to back up, so this is the top 10 selling drugs in the United States. And so and shown in purple are the number of people who benefit from taking the drug. Shown in blue are the number of people who do not benefit from taking the drug. So the best drug up there, 1 in 4 people of those who take it actually benefit from taking it. And in the worst case, 1 in 25 of the people who take a drug benefit from it. So when 24 out of 25 of the people who are taking a drug do not benefit from it, that's a big problem for the healthcare system. So it's kind of nice for pharma, but
this is not the whole population of the world or the U.S.
This is the U.S.
But it's for whom the drug is prescribed.
For whom the drug is prescribed, people who take the drug. So and they've actually started having a requirement that you have to be able to ask your doctor what is the number of people, the number of patients that take this drug for which you get one person that has benefit. They're actually required by law now to provide an answer to that question when you're prescribed something. So this is a serious issue. And so we have many individual differences amongst us. So one of the things that we are advocating at ISB and certainly pushing forward is the notion of generating what we call personal dense dynamic data clouds to try to understand more about what's happening in an individual's body. And we call these personal because they're related to the individual, dense in terms of having a large number of measurements, dynamic because we're following them over time, and generating it that way. And this is what we see as the foundation of trying to build out precision medicine. Of course, there's many initiatives across the country and the world that are aiming to do this. So the way that we talk about this at ISB, this is a terminology that was coined by Lee Hood, but basically around P4 medicine. So medicine that is predictive, preventive, personalized, and participatory. Those are mostly straightforward. But participatory basically means trying to empower individuals with data, with knowledge, so that they can make choices that positively impact their health to the best way possible. This breaks down into kind of two big areas, demystifying disease and quantifying wellness. And we see this as basically the foundations of two industries, one being disease industry, which is essentially current healthcare. I'll talk about that in a moment. And another which we refer to as the scientific wellness industry. And I'll describe that more in a moment as well. So if we look at healthcare costs in the United States, it turns out that 86% of healthcare dollars today are spent on chronic disease, as estimated by the CDC. And if you look at a person's lifetime health over the course of their a person's lifetime health, it turns out that you can attribute about 30% of a person's health throughout their life back to their genetics, sort of the hand they were dealt. About 60% is attributable back to behavior and environment. And it turns out that only about 10% of a person's lifetime health is attributable back directly to the healthcare system today. Now that's a serious issue when you consider that we spend about $4 trillion a year in the United States alone on healthcare. And of those dollars, 97% of them are used on care after illness. And so that's a big chunk to focus on 10% of health. We also have a wellness industry. The wellness industry, of course, is focused on trying to maintain health, but there is kind of this divide between, well, let's say the wellness industry is kind of a mixed bag at the moment. There's a lot of great things in the wellness industry that have a big impact, and there's a lot that don't have a deep scientific basis. I'll give one brief example. I gave a talk at a meeting that had, kind of more of a wellness meeting, and one of the people came up and said, "Oh, we have the best probiotics in the world." I'm like, "Okay, how do we know this, or what's going on?" He says, so I said, "All right, that's great. Like, what is it that's good about these, right?" A kind of a typical follow-up question. He's like, "Oh, we brew them for 2 years until the enzymes are just right." And I'm like, "Okay." Like, these things double about every 30 minutes, so I'm a little confused about what would matter on this time scale. So I said, what happens? So if you measure the microbiome and then you give people these probiotics and then you measure the microbiome after, like, what happens? Sort of no answer, and he kind of leaves. I then look up this company online a little bit later, and it turns out that the secret that they advertise to people is that during that whole period, they play Mozart to the bacteria. And so I'm like, oh. Yeah, exactly. So I was like, okay, we're not speaking the same language here. So this is why we kind of coined this term of scientific wellness, because the term wellness is, we were trying to think if there was a better word for it, but we do think it's really important that we have this focus not just on this care severely after illness, but trying to move upstream. Upstream of that, so that's wellness, but a scientific wellness, which is, can we apply the same kind of scientific rigor in the wellness space that we apply in the disease space? And so this is where we see this rising as a new industry that we call scientific wellness. So the way that we're pursuing this notion of scientific wellness is in two ways. So one is this company that I mentioned at the very beginning called Aerovale. So Aerovale is a consumer-facing company. It takes data of the type that we're going to talk about for the rest of the talk today, and basically it tries to extract information from that to provide a program for improving a person's health. One of the things we realized when we were setting out to generate this huge data set that would give us kind of the ability to see the early warning signs for all the major human diseases, to try to predict them and prevent them, was that the only way that people will stay with you or you'll be able to have an impact or an interface with them is if you have an impact in their life today. And so this was kind of this notion of scientific wellness, so people would stay with this. And so we launched a project, which I'll talk about in a moment, and then after that project, that led to the founding of a company called Aravale. Ed Lazowski actually serves on our scientific advisory board there. We started it a little under 3 years ago. We've raised $54 million for it. Well, actually, that changed yesterday. We've raised $65 million for it, but, but basically, so we've raised money for that, and then we have about 180 employees there now. The other thing that we've done is that ISB has now become an affiliate of Providence St. Joseph Health. And if you're not familiar with this, Providence is the third largest healthcare system in the United States, nonprofit healthcare system. So it's 50 hospitals in the western United States. Lee Hood has now become their chief science officer, and so ISB is now, in effect, a research arm for Providence. And so what that has given us is access to 30 million patient records. It gives us an ability to reach out to a patient population of about 13 million people. So there's an ability to reach out into the healthcare system in a way that we haven't been able to do before. So that's just kind of laying the groundwork for what we'll talk about today. So, so all of this started with a project that we called the Pioneer 100 project that was co-led by Lee Hood and myself. And this was published in Nature Biotechnology last year. And so basically what we did here was when we started out with this intention of generating these personal dense dynamic data clouds for a large number of individuals, and we wanted to prototype this. So we ended up getting, it turned out to be 108 people. So we kept having people asked to be included, so we increased it a little from the target of 100. But it was 108 people, and essentially what we did, oh shoot, I'm missing that slide. Anyway, what we did is we measured whole genome sequences on everybody, and then multiple times per year, so 3 times during the year, we did proteomes out of the blood, metabolomes out of the blood, about 150 different clinical labs out of the blood. We did microbiome from stool, and then wearables like Fitbits and things like this for continuous tracking, along with questionnaires and clinical data and so forth. So we got all that information together, and that's what we refer to as this personal dense dynamic data cloud, or a PD3 cloud. And so that was an experience. Ed Lazowski was actually one of the 100, as he's been very open about, so we don't reveal people's names unless they do themselves. But so in that case, so we took people through an experience, and so we paired them with a health coach that gave coaching advice and tried to have an impact on their health, tried to say that this would have an impact on them, and then generated the data from there. So in terms of outcomes, this is just looking at clinical labs, so it's over a short period of time. But we saw an improvement in nutrition by 21%. This is just based on standard clinical ranges. I don't actually think standard clinical ranges are going to last terribly long. I think that they're going to be modified by genetics, and actually these data are going to let us change them, I think, pretty strongly in the years to come. But in an initial study, that's what we used. So improvement in 21% in nutrition, inflammation by 12%, diabetes by 33%, cardiovascular by 6%. That tends to move slowly. For those people who actually stayed on an Aravale, that's now bumped up to 20% improvement in terms of the number of people who are out of range. For diabetes, just to look a little more deeply on that, for insulin resistance, we actually saw a reduction of 55% in terms of the number of people who were out of range. For nutrition, this related to a number of different things. We can use Ed's example of having very high mercury in the blood because of a like of tuna sushi, which then switched for salmon sushi and then kind of normalized that. So those kind of things, just to do that. Then just one final example. For the people that went through the 100, we had one individual who had basically he was having cartilage breakdown in his knees and ankles, so he couldn't go and do things that he loved with his family, couldn't hike out up in the mountains like he liked to. He had been given a drug for arthritis, which hadn't really been working for him. He had concierge medicine on both coasts. He came into our program. We did this 360-degree view. In his case, something kind of simple jumped out, which is that he had really high ferritin in his blood and a lot of iron, basically. And it turns out, and then we looked in his genome, and he had the genes that gave the highest risk for hemochromatosis. And so we referred him into the medical system, and he, in fact, was diagnosed with hemochromatosis and treated. Does anyone here know the treatment for hemochromatosis? I'm sure Ed does. But. Yep, you donate blood. So if you donate blood, you get rid of that excess iron, and you can stabilize that. So this person did that over the course of 3 months. He was actually able to stabilize the amount of ferritin in his blood, the cartilage breakdown, which turns out that a kind of a lesser-known side effect of this high amount of iron is it can catalyze cartilage breakdown. Its main line is that it leads to liver dysfunction and ultimately death if it's left untreated. But basically, you can get this. And so this was what turned it around for him. What's interesting there is, and you'll notice here on the slide here, that if you've got 0 copies of the variant, it tend to be low. If you have 1 copy, it's higher. If you have 2, it's higher. You'll see the n there is small. So we only have 2 individuals who were homozygous here. So we're not learning this pattern, right? This is an already known pattern. But what's interesting is that the other person there is a woman. She's premenopause, so for obvious reasons, she wouldn't have transitioned into hemochromatosis. But what this does is she gives her information. It says, okay, you've got the genes, you're at high risk for this, but all you have to do is monitor your ferritin levels. If they ever go high, make sure you donate blood. And by doing that, you essentially have eliminated a disease trajectory from her future. And so this is what I think is powerful about this example, is because you can empower people with knowledge to make a very simple choice to eliminate disease trajectories. And so we already know today that you can, by information, reverse disease, you can eliminate disease. It's just a question for how many of those we can do it for. Oh, here's my missing slide in the wrong place. Okay. Okay, so now I'm going to switch to the main topic for today, which is to talk about analysis of the data. So here, I'll start with the Pioneer 100, and then I'll talk about what we're doing on the 4,000 people we have through the Aerovale program. So here, this is from the Nature Biotechnology paper. We did what many people would probably do as a first pass, which is just built a large correlation network. So here, we had metabolites, clinical labs, lifestyle traits, microbiome, genetics, and proteins. You might notice that the genetics there in green is a pretty small set. Obviously, with 108, you're not looking at individual genetic variants. So what we did was take genetic variants across the genome that were associated with different diseases, and then we did dimensionality reduction by just coming up with a number of disease risk scores. I'll talk about them in a minute. And so we basically collapsed the genome down into 70 variables for the purposes of this analysis. Now, what I want you to just think about briefly is that all of these kinds of data had never been measured from the same population simultaneously. So one of the things, and I doubt I'll get much argument about that in this crowd, but when I give this, when I talk to physicians, we often get into this whole thing about not measuring too much about people. But from a data science perspective, if you consider how much we have a large p small n issue, this is 100 individuals, right? A pinprick of a number of individuals, for which we measure all of these things. It's this is the first time that all these things had been measured in a population. And if you just think about the scale of what we're trying to understand from a data-driven perspective in the body, this is the tip of a very big iceberg. But we think it's really critical to try to generate as much of these kind of data as possible. And I'll dive into some of this here in just a minute. So the second thing we did was just to try to identify modules in the data. So here we were just using an algorithm, which is just calculating there in the background. But basically, it just takes from every node to every other node, you do a shortest path, and then you're finding high areas of betweenness, basically, in the graph. So it's like if we're looking at neighborhoods in Seattle, you're cutting the bridges and kind of leaving the neighborhoods that are close together at the end of this. So you do this in order to identify modules. And here's just a zoom in on a set of those. I'm just going to talk about one thing here, which is this connection that you see here from cystine to inflammatory bowel disease. Now, in this case, no one actually had inflammatory bowel disease, other than Larry and his previously. But basically, what this refers to is actually not the disease, but the genetic risk for that disease. And that was really an important thing. So when, at the beginning of this, this started with a vision from Lee and I that we published called the 100K Wellness Project. the notion of getting 100,000 such individuals so that we could watch the trajectories into all the different diseases. And when I was thinking about that number, I think it was kind of like a just kind of imagining just all these different paths, and you want to have enough people kind of come into that bucket where you could try to understand what was happening there. But what we realized early on was that we could actually learn a lot in a short period of time with much less data by trying to understand the genetic risk for disease and using that essentially as the rubric on which we would try to understand the data. Because what that does for you is it means that, while not everyone gets a disease, everyone has a genetic risk score, whether it's high or low for a disease. So it means that every person's data becomes relevant to the study of every single disease. And so that was really a big shift for us. And I'll just show a simple example of that. So one of the things that we found was that there was a strong negative correlation between a person's genetic risk for inflammatory bowel disease and the amount of cystine that they had in their blood. And it turned out, when we looked in the literature, that in fact when people do case control studies of people who have IBD and who don't, you see this same relationship, that cystine tends to be suppressed. And so what was interesting there is that there's actually a mechanism that is suggestive here, which is that cystine sits upstream of glutathione pathways, and glutathione is what combats oxidative stress in the body. And oxidative stress is the trigger for IBD. So immediately, what is suggestive is that these genetics, for whatever the reason, they lead to a depressed amount of cystine that impedes your ability to combat oxidative stress through this pathway. So over the decades of your life, you have less ability to withstand oxidative stress. You're more likely to go into a state where you have surpassed your resiliency and thus might trigger into IBD. Now, if that's true, then it suggests a way that you can change someone who's at high genetic risk for IBD into someone who's at low actual risk for IBD by providing them what they need to make that pathway function at a high level. And so this is a shift from trying to think about it only in the disease state, but you're actually trying to optimize their wellness state by optimizing their resiliency or their ability to deal with oxidative stress, which is compromised, apparently, because of the genetics. So that's what these kind of data are suggestive of. We've got to do studies, of course, to validate all of those things. But in essence, whether or not this particular example holds up all the way to the end, the general principle, I think, basically has to be true, which is that your genetics have to manifest in the body in some way. The more broadly you make your measurements, the more likely you are to see what that is. And I think there's a huge amount that we're going to be able to learn about the states that lead to disease by understanding what's different in the bodies of these individuals as a function of the genetics, the differential risks that we have for disease. So all of those huge GWAS studies that we've done as a community over the past couple of decades can really be leveraged, I think, in big ways with these kind of data to understand those dynamics. And we estimate that there's probably about 60 diseases for which you can make a pretty high quality disease risk score today. All right, so let's shift forward. So now I'm going to shift from talking about that initial study of the Pioneer 100 to talking about what we've done with the data from Aravale. And so for people who come through the Aravale program, as I mentioned, if they check a box saying that their data can be used anonymously for research, then we're able to mine that for discovery and try to figure out ways that we can help do that. And we've been very grateful for this because so far the people who have come through Aravale, about 95% of them have consented to let us use the data to help improve health for them, their kids, their grandkids, try to make discoveries. We're very, very grateful for that. Okay. So we can look at a lot of different disease transitions. This is just a table that I took out of one of our recent grant proposals. But basically, this looks at data from Aravale for people who have had transitions into cancers. And so you can see that this is already starting to happen. So we can look upstream of what happened in the cancer diagnosis. And that's where this scientific wellness approach is really useful because it really lets us get a focus on having longitudinal data that can precede when we make diagnosis. There's lots of data after diagnosis, right? When we and everyone else writes our NIH grants, right? We collect a lot of data on patients, but we don't have, for various cost reasons, typically a lot of data beforehand. So here, kind of the model that's in our head is there's this wellness state. There's going to be some point or short period of an initiation or a transition when you start initiating this disease cascade. There'll be some sort of progression where you affect one network and then more and have a cascade of events. We think kind of late-ish in that stage is when you start having clinical signs, when it might be picked up, and then go to the end. And I'll give a couple of just really early examples of that. So here's one woman who came to the program. We'll refer to her as Eve because she's the first one that we studied. So that's how we refer to her. But basically here, she was diagnosed with stage 4 pancreatic cancer. If you know anything about that, that's a devastating diagnosis. It's an awful cancer. But basically, we went back, and Bert Vogelstein, who you probably know, the world's most cited cancer researcher, he estimates that once you have a diagnosis of pancreatic cancer, you've probably had it for 5 years. So we think she had this actually coming into the program. So what you see here is just a histogram of a particular protein, and it just shows the level of this protein on the x-axis for every person who had come through the Araville program at that time. So our N here is about 2,000. And you'll see there, shown in red, there are 4 outliers. And those 4 outliers are all this particular woman. And so we were intrigued by that, so we looked into it to say, okay, well, what is this protein? And it turns out it's made primarily in the pancreas. It's related to pancreatic cancer. It's in the Notch signaling pathway. And so basically, it's suggestive of something that maybe was an early warning sign for her. Now, that's a long way from being a biomarker. We don't know if other people would show this. And I'm going to talk more about that in a minute. But it's something that jumped out as being very unusual in her particular data. Okay. We don't have to look only at individual analytes. Oh, and maybe I'll say just a couple more words here. So this is just one example. What we've now done, I don't have a slide on it, but what we've done now is built a system where we identify metabolites or proteins or whatever analyte it is that are divergent, outliers on all these distributions. And so we can spit out for every single individual, here's everything that you have that's really unusual relative to a wellness state. And I'm going to come back to that more in a minute. We don't have to do that only for analytes. We can also do it on pathways or networks. We use one particular approach we've adopted, was a method that Don Gieman at Johns Hopkins and I invented a few years ago called Dirac. I won't get into the details of it, but basically it uses as a proxy for the state of the network just a rank ordering from the most abundant to the least abundant of all the components in that network. And then you look for kind of shuffling of that ordering as a sign that that network is in a perturbed state. There's lots of ways you could do this, but this is one way that we've done it to start with. And when we did that, it turned out that a network that was very highly perturbed for this woman was the TNF signaling pathway. This is not in a tissue. This is just the portions of this network that are reflected in the blood, and looking at it in this kind of simplistic way, but just the rank ordering, turned out that by that statistic, it came out to be highly significant that her TNF signaling pathway looked to be highly perturbed. And you can see that kind of in advance of the diagnosis. We then did this, and this is a little bit of a crude analysis in the sense that we're building this out, but we went into the KEGG networks, and there were 9 pathways that we identified where we had reasonable coverage of things that were in our blood panel. And our blood protein panel is about 300 proteins. So we have a new set of data that we've just done that will give us 1,000 proteins. And then we have another set of data that we're in contract discussions right now, but that will give us 5,000 proteins. And so we're extending that out. But just on the 300, there were 9 pathways that we thought we had good statistical coverage for. And so what's shown here then is this just says how many of these pathways were divergent, so highly different from normal. We'll just call that divergent. And so here, so you can see 670 of the 948 clients that we analyzed at this point, there was 0 differences, 140 had 1 that was divergent, and so forth. And here what's shown in the visits, these are the visits that preceded Eve's diagnosis of pancreatic cancer. And so we just looked into this. And so visit 1, so this would be 18 months before diagnosis, we saw no sign in the networks. We did in the individual analyte, but not in the networks. And this is only 9. We're working this out so that we'll have much more networks. That's just my research scientist, really great scientist, Noah Rappaport's working on that right now. But anyway, what happened was that, so 18 months out, there's 0. 12 months out, there's 1. 6 months out, there's 3. And just before diagnosis, in visit 4, there's 5. And so what we see is like this is kind of our first look at what might be a cascade of networks that are just going out, that are getting perturbed as you go along. The resolution here will be a lot better as we work out to more networks, but what this system is going to let us do is actually just monitor and observe network perturbations dynamically in humans. And we can do it with quite a lot of fidelity. So that's what we're trying to build out right now. These number of outliers, or these divergent values or atypical values, are they increase with age. So this is another interesting plot. So this comes out of John Earle's thesis, who's a graduate student in UW Computer Science that works in my group. But basically here, this is people who are in their 20s all the way up through their 80s. So this is age on the x-axis. And what we see, this is the number of outlier values that they have in their data on average. And you can just see that as they go from their 20s, 30s, 40s, 50s, 60s, 70s, 80s, and so forth, that it just goes up monotonically on average. You can see the error bars here are very large, so there's a lot of room for individual differences, right? So someone in their 60s may have fewer of these than someone in their 40s. But on average, this just sort of ticks up with time. What was really interesting is we tested this to say, in this case, we were counting as outliers things that were in kind of the extreme, I think 2% of the distribution. We tried it actually to make it, just to try to learn, we tried it if we said it was at 5%, the upper 5 or lower 5. We did it for the upper 10, lower 10. We even did it for the upper 20, lower 20. And what was amazing was that this pattern persisted no matter how wide we made the call of divergent. What that told me was that there's already starting to be really subtle regulatory changes. So even things that just slightly move, that happens a lot more as people age, at least according to this analysis. So that we thought was quite interesting. So where are we going with this? So I've shown you just kind of little bits of the beginning of this because we're just kind of observing small numbers of these in the initial, but where are we going? So what we're trying to do is to develop a new paradigm for how to think about diagnostics. So traditionally, diagnostics, and we and others have done tons of these, but you usually do a case control, right? You get your cases, you get your controls, you compare them based on age and sex and smoking status or whatever it is, and you get all your cases and controls, and then you figure out what's differential between them, right? And then whatever comes up are candidate biomarkers and so forth, right? That's the typical thing. So here we're going to do something pretty different. So because here we don't have case control, because we don't know in advance like what disease a person might manifest, right? So we don't have like a disease cohort. So everything is going to be defined based on a wellness state, and then we're going to be doing analyses of deviations from that wellness state. Okay, so there's no case control. So the wellness state will be calculated from a population, and there's all kinds of ways that you could think about doing that. And actually, hopefully, we can get into some interesting discussion around that because that's really a challenging and interesting problem. But then what we'll do is look at divergent values on an N of 1 basis for the individual. So you then take, so for every individual, this is the system that we're building out this year, and it says, okay, for you, here's every analyte or every network that's in an atypical state relative to that wellness state. Okay? This can be informed by dynamics. In other words, we can monitor it over time. Is it becoming increasingly out? Is it bouncing out and in? I didn't show this, but we actually have calculated what we call transition probability maps for every analyte, so we know how often they tend to pop into atypical states and back for every analyte across all the thousands of people, and we can compute how unlikely something is that we observe. Now, the really challenging part here, is to then do a functional analysis of individual divergence. So essentially, you have to build, I call this our context interpreter. This is aspirational right now. We're trying to figure out how to build this. But basically, the input is this vector of everything that is unusual in an individual, and the output is, right, tell me what that means. And so what's inside that algorithm, it's a lot of systems biology, it's functional annotation of what these genes do, where are they located in the body, et cetera, right? So it's as much knowledge as you can put together to try to get an interpretation of does this look like early stage pancreatic cancer or not, or, or whatever, or does it look, is this a bad blood sample? Is this, like the universe of possibilities gets large. And this is where we'll have to get into, AI, machine learning, systems biology, deep context learning, all that kind of good stuff, because this is where the problem gets most interesting and most challenging. But what's really interesting about this kind of an approach is that this would enable truly personalized medicine, because this kind of an approach, unlike pattern learning or molecular signatures or things like that, which I also do a lot of, but what this would do is this would actually let you make a diagnosis on a pattern that you've never seen before, because it was a functional analysis of what's happening in that person's body. So that's why we're really passionate about this kind of an approach, despite the difficulty of that last step, is because this would be actually a path to truly personalized diagnostics. And so that's where we're trying to get to. Okay, let's see, how am I doing here? We've got 11 minutes or so. Okay, so I've got 2 more stories to tell you. So one of the things that we've done here, and this is also work from John Earls, but this is looking at metrics for, what we're trying to develop things that we call metrics for wellness, or trying to think about things not only in the disease context. And one of the simplest things to do as a first pass on this is calculate biological age. You've probably seen these kind of calculations in a couple of places. Probably the 2 most prominent that have done this down at UCLA, Steve Horvath has done a lot of this with methylation data, and then we had him up to ISB a couple of years ago, actually, around that. And then Joel Dudley, who's a good friend and colleague out at Mount Sinai, has recently done this from electronic medical records. So we're doing something similar, but from the data clouds. And so in essence, what you're doing is you're just saying, if all I knew was the proteome and the metabolome and the clinical labs and so forth, can I predict this person's age? Okay, so that's the first step of it. And then what you're trying to do after that is then you kind of ask the reverse question, is if I just say, how old do you look based on these measurements, you say that's your biological age, and then is that biological age somehow better predictive of your health or your health outcomes than is your chronologic age? So John has developed a method to do that. There's a great algorithm for this that was developed by Clamara and Dubal that gets rid of bias in this. In other words, you kind of want a biological age that has a slope of 1, so it's a little more, because most of these biological ages, actually all the other ones that are published, they have a slope such that biological age only goes up by like 0.7 per year or something like that. And so your young people always look older and your old people always look younger. And so with this algorithm helps us to come up with a fit that is that doesn't allow that. And so it basically gets rid of that bias for the most part. So the real question, though, is this related to health? We're just at early stages with this, so but I'll show what we've done. So all we did is take our data and just put a few things in there that we thought should affect health one way or the other, and see, does it move this biological age the way that it should? So if we do that, so we just, a really easy one was, does someone have type 2 diabetes? That's highly prevalent, so we have a lot of cases. This is a complex plot, because I haven't simplified it down from the thesis yet, but basically, the red line here, this just shows all the various different kinds of algorithms and different types of data. So these are all the different models that were run. And this just shows that in every case, the red line being above 0 here, just shows that, yes, type 2 diabetes under every model that we tested, it always increases this biological age. So that's just a sanity check. Makes sense. Coronary artery disease, same thing, increases biological age. Then we said, well, what about Fitbit? If people use their Fitbit a lot, does the biological age, is it low? Yep, it is. And so we have some sanity checks on that. So we're just exploring that in a little bit more detail. And what we really want to see is, for an individual, how meaningful this can be. So as people make changes in their lifestyle, for example, can they actually have an impact on this biological age? And then does that show something related to health is what we're working on there. So this is kind of our first pass towards a metric for wellness. Okay, and then the last thing that I'll share in terms of data is a case study here. This was done primarily by Andrew Magis, who did his PhD with me, but is now director of research at Aerovale. But this comes out of the data, and I think it's quite interesting, which is related to statin usage. And so statins are the most commonly used drug in our population, at least. And so it's one where we have a lot of statistics here. We get good numbers. So you can see kind of for the different labs, number without statins varies between 700 up to 1,500 depending on the data type. And the number with statins is typically on the order of 100 to 200 or so. So we've got pretty good numbers there. So what we wanted to do was just to select phenotypes that were associated with statin usage. And so here, you'll see that the number one metabolite that comes up is HMG. And you can see kind of what the distributions look like here. But this one actually makes a lot of sense. You can see the p-value there, right, was 10 to the minus 15th, so very significant. This one makes a lot of sense because HMG is actually a substrate for the enzyme that's targeted by a lot of statins. But what's interesting here, so statins will come in and inhibit this. And so if you're on a statin, this metabolite really builds up a lot relative to not. The usefulness of this is that it could actually be used as a proxy for statin efficacy. So as I showed on the imprecision medicine slide, a lot of drugs don't work for a lot of people. And so if you're on a statin and it's not really affecting your cholesterol, then what this will tell you is that if you look at your HMG levels, then that would probably tell you whether or not the statin is in fact hitting its target. So it can help you differentiate between, is the statin hitting the target, but you have some compensatory mechanism that's making up for that cholesterol level, or is the statin you're on actually ineffective at getting the target? So this metabolite will tell you the difference between those scenarios. So that we thought was pretty interesting. You also can get new associations in the data. So this is basically just showing a reduction in the degrees of freedom, in a sense, when you take a statin, in the sense that here, HMG, in the normal population, there's no correlation between HMG and cholesterol. As soon as you take a statin, then in fact, there does become a correlation there because the statin has now tied those things together. And so there's a lot of examples like that. Here's one that I think is very interesting, which is that in our data, we found hemoglobin A1C, insulin, and glucose are all associated with statin usage. And if you know anything about these clinical markers, these are all the clinical markers for diabetes or prediabetes. And so you can see that the taking of statins has an effect here. And in fact, another group had done a meta-analysis looking at actual disease outcome, and it turns out that if you take a statin, there's a 9% increase in the incidence of diabetes if you do that. So what's interesting about having these kind of personal data on yourself is that with the markers, you can go in and say, okay, is the statin actually having an effect for me on diabetes? And one of the things that we're really interested in is trying to build maps for everyone around your disease proclivities. In other words, how likely is it for you that high cholesterol is really a bad thing, and how likely for you is it that diabetes is worse? And those kind of relative risks, which are not dealt with in a highly personalized ways typically today, at least not really heavily data driven. So those are the kind of things that we're trying to work towards. So I'll just say just a few things about a few other big projects that are ongoing. I already mentioned this affiliation that we have with Providence St. Joseph Health, which we're excited about, with Lee becoming Chief Science Officer over there. But what this lets us do is to launch a series of clinical trials as well. So as I mentioned, we have this wellness cohort, people coming in through Aravel, so we've got thousands of people where we have data from them. And then we have disease-specific cohorts as well. So first, we are doing a trial around scientific wellness. So this is Providence has put, Providence St. Joseph, I should say now, has put 1,000 of its employees through the Aravel program. And then they're doing an analysis of those 1,000 people versus a control population, and basically seeing what are the difference in health outcomes, what's the difference in economics, and so forth. So that'll be a 3-year study to look at that. We also have launched a clinical trial around Alzheimer's disease, and we are collecting these dense dynamic data clouds for individuals who have mild cognitive impairment, and then testing more lifestyle kind of interventions for them to see if there's an impact. And actually, and I spoke at Genentech recently, and the first thing that their head of neurology said to me was, was very interesting because they said I thought I was going to fight them on this point, and they said, well, so far, no drugs have shown any efficacy, and only lifestyle interventions have ever had an effect in Alzheimer's disease that's been proven. That's actually true. I said, oh, I thought I was going to have to make that point. She's like, nope, we already agreed. So that's been true. So we are testing that out in a set. But the other thing we're really doing is trying to look at stratification with the dense dynamic data clouds to get insights into things that we might be able to do for Alzheimer's disease with multimodal therapies and things of that nature. Let's see, I don't have a slide on this one, but we also are just about to finalize a contract with Genentech around a new study where we're going to do the dense dynamic data clouds for multiple sclerosis patients, looking at fast progressors and slow progressors, and again, trying to understand the landscape of what's happening there. And then the final one I'll mention is we've also launched a clinical trial with Swedish Cancer Institute on breast cancer survivorship. And essentially, this is an effort. We are very happily having more and more breast cancer survivors, right? The treatments for breast cancer have gotten much better. Personally, I'm extremely happy about this. My two little sisters already have had breast cancer, so my youngest sister got stage 4 metastatic breast cancer at 23, which is the youngest case I've ever heard of. Yeah, crazy. Got great treatment at UPenn and is doing great. But one of the things that happens is that just going through the chemotherapy and radiation has a big negative impact on health. And there's all kinds of issues that come along with that. So we thought that maybe the scientific wellness approach could actually be useful in the context of people who have survived cancer, but trying to get them back really, back to full health. So we have a project where we're going to take 100 women through this program with Swedish Cancer Institute and prototype this, and also just try to get a molecular basis for all the kind of things that happen to them, some of which are kind of vague. All right. Conclusions. Anyway, some of which are kind of vague, like brain fog and things like this that they talk about. But can we understand something more molecular there? All right. So to conclude, scientific wellness we really believe is going to be the key to disease because it gives us a framework in which to see the earliest disease transitions. We think signal to noise is going to be a lot better there before this huge cascade of things have happened, so you can actually understand something about trajectory, and especially about what individual trajectories into disease look like. So those early transitions are going to be really essential. There's many ways to do that. I talked about two of them, the single analyte and the network perturbation. I didn't get into context-specific deviations, but those are very interesting as well. And yeah, these personal dense dynamic data clouds we think will be the center of building out P4 medicine. So with that, let me thank people in my lab. I think I thanked most people along the way. But I think, so Andrew Majors, John Earls really were the computational drivers for the Pioneer 100. Gustavo Glusman contributed immensely to that as well, especially around genomics. And Noah Rappaport did the network analysis models that I showed as well. And happy to take any questions. Thanks. Yeah.